Good afternoon to everyone. I know this is post-lunch session and everyone wants to doze off a little bit, but I hope to keep you all awake by presenting an exciting story here. So I'm actually going to present a recent development in the functional genomics of the malaria parasites. As many of you know about the malaria impact, but I just wanted to refresh your memories. And malaria is a vector-borne infection which is transmitted by mosquito, and half of the world's population is at the risk. And every year there are like 200 million cases reported around the world. And 90% of these cases actually occur in Africa. And there are like 500,000 children die of this disease. So basically, if you make a math, one child is dying in Africa every minute. Now you can realize how important is this disease. As far as the life of these parasites are concerned, this is transmitted by the infected Anopheles mosquitoes. And so when the infected mosquito bites a human being, it deposits sporocytes, infective stage, and these parasites get into the liver through bloodstream where it develops rapidly and releases merocytes, and these merocytes enter into the fresh RBCs where they undergo some asexual development again, and this goes for a few cycles, and then after several cycles, and some of them becoming gametocytes, and wait for another mosquito to come and pick it up, and then rest of the developments occurs in the mosquito midgut, and then the cycle actually continues. I just want you to focus on the very important stage in human infection. That's actually the blood stage, because this is the stage which causes all the classical symptoms seen in malaria patients. To give an overview of treatment and control of this infection, I know there is a recent development in the malaria vaccine development. Like last month, I don't know how many of you know, last month Europe has actually approved use of the first malaria vaccine called Muscurex. However, the efficacy of this vaccine is low, around 30 to 40%. So chemotherapy is the gold standard for treatment of this infection. Earlier days, chloroquine was used for treating this disease, but the resistance is rampant nowadays. But we have, luckily, artemisinin combination therapy that is very effective against this infection. However, we are recently seeing resistance to this drug as well. So it's imperative to have new drugs developed for treating this infection. We do have a potential because there are 50% of the genes that encode proteins which are unique to the parasite. And the functions of these proteins are unknown. So we can actually explore these genes and come up with a new treatment. But there are challenges. What are the challenges? This is very difficult to do manipulation. As you saw, the malaria life cycle, it's a very complex life cycle. And also the genome is very AT-rich, so you cannot do any clone. You can do cloning, but it's very difficult. And the second challenge is there are no reliable gene regulation tools to study the biology. To talk about the gene regulation in these parasites, so the siRNA machinery, which is a powerful tool used in the mammalian cells to study mammalian cell biology, is not useful in the parasite because the siRNA machinery is absent in these parasites. And then we do have some regulatory tools such as post-translational regulation system, and we use the destabilization domain to do this conditional knockdown in these parasites. One such destabilization domain is called FKBP, but there are some disadvantages. One is actually it requires very expensive material called Shield 1. And then it doesn't work for most of the organelles, such as mitochondria and apicoblast-targeted proteins, and also the membrane proteins. Membrane proteins are really, really challenging to work with. Given the focus of our lab, which is developing new tools to study malaria parasites, we have come up with a new idea to regulate the essential genes. What we call is as TETR aptamer system. So basically, this is a post-transcriptional gene regulation system. You may or may not have RNA, but there will be no protein. So this system is consists of 3 different components. One is the TETR. TETR is the translational repression protein seen in bacteria. Second one is the RNA aptamer. So as you can see, aptamer is an oligonucleotide or peptide that can actually bind to a specific target. And then a small molecule called anidotetracycline. So the way the system works is like you actually put the RNA aptamer between the promoter and coding sequence and then overexpress this TETR protein. This is bacterial protein, so you're expressing in a eukaryotic parasite. And then this TETR binds to the aptamer, thereby blocks the ribosomal binding and hence prevents the translation. The minute you put the ATC, the TETR binds to ATC rather than aptamer. So now the transcripts are free. Now ribosome can bind to the RNA, and then translation is happening. So this is how you on and off the system. We actually tested this system in the basic eukaryotic system called yeast. And basically, it worked well in yeast. And then we moved on to do in Plasmodium. And we come up with an idea to use firefly luciferase as the reporter system because you want to see how well this works, and also you want to study fold regulations. So based on our studies, we found that there is 5-fold difference between plus and minus ATC conditions. This is really good because we have something that works for regulating genes. But if you could see the minus ATC thing, so you can actually see there is still some protein expression. This may not be good for studying some of the proteins, especially the enzymes, because even the basal level expression could be good enough for the parasite survival. So we wanted to improve this system better. We have come up with another idea. Basically, this is the TETR system. Now we have, this is actually unidimensional because it completely depends on just TETR binding to the aptamer. But if you look at another direction, so we made some TETR fusions. And basically, this TETR fusion could be some RNA effector things because we want to change the RNA dynamics. So we can basically use the RNA effector proteins and fuse it to the TETR, and still we should have the regulatory activity. And when we look at the literature, so basically these RNA effector molecules may do decapping, deadenylation, or RNA sequestration, which can further actually regulate the protein expression. So when we looked at the literature, we found a protein called DOSI. This is actually DDX6 family protein. It's very well conserved in all the eukaryotes and involved in variety of RNA functions. So, and most importantly, this has been shown to be involved in the translation regulation in the parasite. So we put this gene to make the fusions. And then to test the system, we actually put all the regulatory elements and the aptamer and then the target gene as a reporter gene fluke in the same plasmid. And then importantly, we put aptamers in differ' positions because these proteins, the DOSI protein can mostly work on 3' UTRs. So we tried that. And also only at the 3' end. So basically we made 3 constructs for TETR and 3 constructs for TETR DOSI and then transfected into the parasite. So these are results from our luciferase experiments, dual luciferase experiments. And as you can see, when we had only 5' aptamer, so we still saw regulation with the TETR DOSI. That's good because we still preserve the regulation, but there is no improvement. Interestingly, when we saw 3' aptamer alone, so that the red one is actually mutant aptamer. So basically the TETR protein won't bind to the mutant aptamer. So when we did the 3' aptamer alone, we were able to see no regulation with the TETR, but with the TETR DOSI, we were seeing 20-fold regulation, which indicates that the DOSI fusion is actually getting some gain of function. So it's cool. And then we went on to do like these two 5' and aptamer, 5' and 3' aptamer contacts. We were seeing like almost 70-fold regulation, which is exciting. And so we were excited when we looked for whether this regulation is conserved in all three asexual stages, like rings, tropes, and schizons, and it's preserved. So we were happy. And then we actually replaced the Firefly luciferase with YFP because we want to see whether this is transcript dependent. And interestingly, we saw the same thing with the YFP. So the red one indicates like an induced one, the green one is like uninduced one. So basically, you can actually regulate even the YFP. So it's not transcript dependent. So we can confidently go ahead and study the parasite genes. And we also tested with different promoters, and we got like 200-fold with one of the promoters that we commonly use in the field. Now I'm going to switch the gears. So we know like we have an exciting system, how we can implement this system to study the parasite genes. So there are two strategies based on my luciferase experiments. So one is actually the regulation by 3' aptamer alone installation. And the way to do is actually do the allelic replacement. This is a very common technique used in the field. So basically use 1 kb fragment from the coding sequence, at the end of the coding sequence, and then put the 3' aptamer, and then transfect into the parasite, and then get the integration. So now you have this aptamer installed at the genomic locus. The second step is like you overexpress the regulated version in a non-essential gene locus and then knock out the endogenous locus, so that now you can actually knock out this endogenous locus and then regulate with the ATC to see whether this gene is actually essential or not. So we have used both the methods, but the time shake, I'm not going to show everything, but I will be showing one method, which is allelic replacement method. I picked a gene which is very important in the field. That's actually P-type cation transporter ATPase, because this is an important drug target. Believe me or not, this has like so many compounds that acts on this particular target, but people don't know anything about this other than its localization, maybe some putative function. And so like I mentioned, there are several antimalarial compounds that targets this one. And then there is no direct genetic evidence for its essentiality, but these drugs are already in the phase 2 trial and stuff like that. And then this is a membrane protein, so it's very difficult to do like conditional knockdown. So I took this gene, and then I was able to replace it, allelically replace it at the endogenous locus. This is just the schematic showing how the locus looks after replacement. And then I confirmed this by diagnostic PCR as well as the southern blots. They were clear that I actually replaced the original genomic locus. And then I went on to do some phenotypic studies. So this is the parental strain, NF54, where you can see that plus and minus ATC doesn't show any difference. That is good because my tetradosi overexpression is not killing the parasite. Then I picked 2 clones that I'm confident that as the replacement. And now you can see that this is the dotted lines are plus ATC, the continuous ones are minus ATC. You can clearly see that there is a difference in the growth pattern for clone 1 as well as the clone 2. And this is actually cumulative fold expansion. That's why it looks like it's still there. But this is like over a period of time. As you can see the difference, it's like 1,000 to 10. So it's basically like they are dying. This is just like plus 80. I merged the plus ATC with the NF54, the parental lines, so they follow the similar trend. So basically, this gene seems to be essential for asexual stages, and so people can continue work on this particular drug target. And this is the first time this has been shown in the field, like the PFATP4 is essential for the asexual stage development. And like I did Western blot just to confirm there is no protein with my minus ATCs. As you can see that minus ATC lane doesn't have any protein, but the plus ATC do have. And so this is my loading control cavity edge. So it's exciting. So in summary, we have a robust system, and it's a powerful knockdown technology that can work for like essential genes. And then it's versatile because you can use like several ways. We can modify the genes of any kind in the parasite. And then it also has potential applications to identify new drug targets. And we are working on several unknown genes in the parasite now so that we can come up with the new drug targets. And then finally, this can be used in other eukaryotic pathogens and mammalian cells because the DOCI or the DDX6 proteins are well conserved throughout eukaryotes. With that, I would like to acknowledge my advisor, Jacqueline Niles, and then Haleha, who did like some critical experiments for this work. And then my collaborators at Broad, especially Nobu, who is currently doing some drug sensitivity assays. And then my funding agencies. I hope with our tool, one day, we can imagine a world with no malaria. Thank you for your attention, and I would be happy to take questions.